in April. And yeah, thank you very much to be here. Good morning. The title, give an outline. First I want to talk a bit about RNA-based predictions of hybrid performance, and in the second part of my talk about what I like to term breeding-assisted genomics, the use of phenotypic data from breeding populations to characterize phenomena, in this case heterosis. Hybrid breeding, oops, it's there. A good example is mice, leads to, in maize, to tremendous increase in yield. You can see here, and it's not only this yield increase, but it's also the higher stress tolerance of hybrid plants, which makes hybrid breeding very interesting. It's a contribution to food security, and there's a large interest in the transfer of hybrid breeding and optimization to other crops like wheat, rice, or rapeseed. Why RNA data? The challenge in prediction of hybrid performance is that we have to use parental information to forecast superior hybrids. So, and when we can forecast, we can reduce field trials, which are time-consuming and expensive. Double haploid technology even increased this problem because it allows to generate hundreds, thousands of new lines in a year. And in hybrid breeding, we have two different populations, and there are immense amounts of crosses possible, combinations possible. So the idea to integrate RNA data was that RNA data integrate information. I mean, transcription factors have to act on the DNA that an RNA is transcribed, or the epigenetic state of the locus. All this contribute to the quantitative measure of RNA data. Usually, best linear unbiased prediction models are used. And here we looked at parental relatedness. We also use a binomial distribution model, which was published first by Frisch et al. 2010. Here we look at parental differences. And the big advantage of this model is that we can identify trait-associated features. But I will talk about this in the second part of my talk. Here, an example of hybrid prediction, cross-validation. You see here, it's a blood approach. The largest we did yet with 270 maize hybrids, and we used for genotyping the maize the 50 chip array, the Illumina array, and for RNA data we used the custom 2K microarray. The traits you can see here are dry matter yield and dry matter content. And what you can see here, we have different test sets of the hybrid. T0 means that no hybrid has parental lines with former test crosses. T1, that one parent was involved in test crosses in this test set, and T2, both parents had test crosses before. So the most challenging is T0, and when you look at this here, the dark green bars, the pedigree information and genomic information leads to quite good predictions, but here the transcriptomic data are the best predictors of dry matter yield. And it's even increased a bit when we combine genomic and transcriptomic data. For dry matter content, so this is the performance of these data is trait dependent. For dry matter content, you see that the transcriptome is not the best, but in combination with the SNP data, it increased prediction accuracy quite a lot. So this is really promising. And why is this? We cannot give a full explanation, but what we did is an expression QTL analysis with this data set. And what you see here are the distribution and relationship of genomic and transcriptomic data. And we see significant association between gene expression and SNPs. These associations are adjacent and on the same chromosome or across chromosomes. So what we think is that gene expression data capture epistatic interaction, and this leads in some cases to an increase in prediction accuracy. The next example I want to give is in rapeseed. Here we had 750 hybrids about phenotyped in field trials. Altogether 12 maternal lines and 374 double haploids, the fathers. We used the 60K SNP array and here RNA sequencing to generate the RNA data. We used messenger RNA data and small RNA data. It's always a problem where to isolate the RNA from, so which sample we take, and usually we decide for seedlings. And here you see an example. We have here controlled growth conditions, randomized trait position. We pool 5 replicates for each genotypes and harvest the entire seedling. We make sure that the primary leaf here is developed. With these RNA data, the prediction accuracy results look a bit more diverse in rapeseed. Here I show you 2 traits, yield and oil content. We use the binomial distribution model, and here it's type 1 cross-validations because all maternal lines, we have only few in Brassica, were involved in test crosses before. I think this is a realistic situation for rapeseed breeding, and we use 25% of the fathers for the training set. What you can see that in one subpopulation this year, the yield or the prediction accuracy increase for yield with transcriptomic data is quite high in comparison to genetic or SNP data. This is not the case, or not that pronounced here in this population. We have for oil content here in this population a large increase. Here small RNA data perform as well quite good. And what we see is that the combination of different data do not increase prediction accuracy here. And we have no increase or no better prediction for transcriptomic and small RNA data in this population for oil content, for example. So the picture is quite diverse. A lot of work has to be done here, and I think the future directions for is feature selection based on functional categories, for example, and deep learning approaches. But now I want to come to the second part. I like the quote, breeding-assisted genomics, using the phenotypes of breeding population to characterize heterosis. This is what we do. Heterosis is the superior performance of hybrids. There are various genetic mechanisms involved in heterosis. I think this we can agree on. And it is clear that it is dependent on the interaction of distinct genomes. They have to be different. And to ensure this, There is the heterotic group concept. Two heterotic groups are bred separately, independently, to maintain the diversity between them. Here, I want to show that heterosis is, we can see a heterosis very early in development. 6 days after pollination, we see in some lines double size of the hybrid embryos due to a higher proliferation rate. So these interactions of the genomes take place very early on after fertilization. Here is the data set I want to talk about in the next few slides for the rest of my talk. It's a systematic study of a 7 times 14 factorial breeding population, dent and flint lines. We have these 2 heterotic groups, 98 resulting hybrids, and the trait we analyze here is mid-parental heterosis for grain yield. For this data set, we have small RNA, messenger RNA, and SNP data, here with all the lines with the red dots, and for 3 hybrids. And importantly, again, here we used RNA data of seedlings, 7-day-old seedlings in this case, and we relate this data, correlate and associate these data with field trials. First, we compared the data types, and we used distance measures. Basically, these measures sum up the differences between the parents. And what you see here is what we expect when the distance or the differences between the parents increase, we have higher heterosis. The same we see for messenger RNA data, with more differential expression between the parental lines, we see more heterosis of the hybrids. But this is not true, and it's opposite for small RNA data. We found here a negative correlation, so the more small RNAs are differentially expressed between the parental lines, the lower the heterosis we see for the hybrids. And this shows that there is a fundamental difference in the relation of heterosis with small RNA data. So they are quite interesting for us, and I want to show you some further results. But first, what are small RNAs? Usually these are RNAs of 20 to 24 nucleotides in length. They are involved in gene regulation. The main mechanism here is RNA interference, and they are involved in the modulation of the epigenetic landscape. RNA-dependent DNA methylation is the best known mechanism here. Importantly, small RNAs are mobile and transacting, so this renders them ideal candidates for managing the interplay or the programming of the diverse genomes in a hybrid. First, we analyzed the variability of small RNAs in our populations, and there is an extremely high interspecific variability. You can see only 35% of the small RNAs we identified are expressed in both dent and flint lines. In total, we have about 600, we talk here about 600,000 unique small RNAs in these 21 inbred lines. The next step was to identify heterosis-associated small RNAs, and here we used a binomial distribution model. So the hybrids are ranked for the heterotic value, and we form 2 equal-sized groups, the low and the high yielding heterotic hybrids. The null hypothesis for this model is that the differential expression in the parents is equally distributed among all hybrids and or among these two groups of high and low yielding. And we test if the small RNA is more differentially expressed in the parents of the high yielding or low yielding hybrids. And we identified in this way about 10,000 small RNAs associated with heterosis for grain yield. We controlled by permutation tests, shuffling the hybrid trait values. There we got no significant associations. And we found different directions of associations. We call them positive. And when you correlate this, again, with a distance measure here, a binary distance, the more differences we find between the parent, the higher the heterosis the hybrids show, and for the negative ones, it's opposite. Importantly, we found more, 64% negative associated heterosis-associated small RNAs. When we look in the hybrids, we see that we have largely non-additive expression. Here, this part of the parental small RNAs, only about 25% are expressed. We found back in the hybrids. These are only 3 hybrids of the whole population, and 10% are new. We didn't see in the parental lines. So we had to look about the heterosis-associated small RNA expression in the hybrids, and if they have functional relevance, we would expect that more positively associated are expressed in the high heterotic hybrid and vice versa. And this is exactly what we see here. But is this cause or effect of heterosis? And to test this, we try to interfere with small RNA biogenesis, and we did this by a transgenic approach. We expressed the gene silencing suppressor of wheat streak mosaic virus, P1, with an inducible expression system. It has a bidirectional promoter, here a marker gene we could express at the same time. We integrate this transgene in the A108 background for 8 generation, and the crosses we did, you can see here, we used hemizygous transgenic plants to cross with the same lines, so to produce the inbred lines, 50% transgenic, 50% wild type, and the same we did with another line, H99. It's due to this line, the choice for this line is due to its well-known tissue culture performance. Because we used an in vitro system to induce the transgenic system. Here we isolated 1 day after pollination, nucellus tissue, grow this in vitro, 50% always with induction, 50 without. It takes about 30 days for the embryo to germinate, and then we did determine the growth rate of the seedling plants. It was a kind of observer-blinded study because in the end we tested if the plants were transgenic or not by simple gas staining. And the next step was to make sure that the P1 suppressor has an effect. And yes, we see here by bootstrap analysis in the hybrids that the complexity of the small RNA population in the induced plants were significantly reduced. We found about 27% reduction. And what we see that here we looked at the conserved small RNA in these lines, that the largest fraction which is downregulated is the negative associated small RNAs. So this fits, and our expectation is that we have an increase in heterosis, and indeed this is the case in the control non-transgenic ones, so control that the DEX induction has no effect. We see no difference, but here when we calculate the best parent heterosis, we see an increase, a significant increase in this P1 expressing plants of about 15%. So a reduction of small RNAs positively influence heterosis formation. These data are limited, it's only one genotype combination, we have to repeat this, but it's, I think, a strong indication that small RNAs are a component in heterosis formation and have a negative effect overall. So, what are these heterosis-associated small RNAs? What we see is that they are enriched for 22 and 24 nucleotide class, and this is good. These are canonical or known functional classes of small RNAs in maize, and we found no association except of one microRNA. So RNA interference seems not to be highly involved in this heterosis influence, or this influence of small RNAs on heterosis. The next analysis was about the genomic distribution of heterosis-associated small RNAs. We see a uniform distribution, mainly here, but what we found is that exclusively the negative 22 nucleotide associated small RNA are enriched in pericentromeric regions. This is true for all all 10 chromosomes of maize. And we have another feature here, the 22 nucleotide negative associated small RNAs map to multiple locations, predominantly, you can see here, and they map to multiple features of the genome. We just divided the genome simply in gene, intergenic, and repeat regions and the intersections of this. And here the green part of the bars shows the negative 22 nucleotide small heterosis-associated small RNAs, and they map predominantly to multiple locations. And this indicates that they might act in trans, originate at one loci and act on another loci. Another interesting feature of these small RNAs is that they are enriched for transposable elements. Here these are two elements with no evidence of transposition in the last 360,000 years. And these elements show predominantly, these ones show spreading, chromosomal spreading. It means that the chromosomal state is not local, but is spread around. So there is an influence on gene expression likely when a gene is close to these transposomal elements. And indeed, we found that the heterosis-associated RNAs are enriched near differentially expressed genes. This is true for the Flint-Dent factorial we used, and as well for B73 Missouri lines. We found an enrichment for gene expression differences, promoter usage, and as well for splicing and enrichment of the small RNAs. And the model, how we assume this pericentromeric enrichment of the 22 nucleotide small RNAs might have occurred, is this. It links genetic architecture, recombination levels, and the 22 nucleotide associated small RNAs suppressing heterosis. In the beginning of a breeding scheme program, we have here in one heterotic group these small RNAs in the pericentromeric regions, and they are usually used for transposon silencing, but they have also negative regulatory effects on gene expression in trans. And breeding direction selection then select against these negative regulatory effects, because we have here low recombination in suppression of recombination in the pericentromeric regions. Only the outer arms of the chromosomes can be recombined to get rid of these targets. And in the end, we have this optimized 22 nucleotide small RNA target combination in invert lines. But when we then hybridize, so combine two genomes of different heterotic groups, these small RNAs from the one group might act on the other chromosomes from the other heterotic groups, find there the targets for negative regulatory effects back. So this is our working model, and with this I want to conclude. I showed you that messenger RNA and small RNAs are capable to predict hybrid traits, and RNA data can enhance prediction accuracy in combination with other data. The future directions are feature selection and deep learning approaches for this. I showed that RNA-based association approaches with breeding populations facilitates the identification of trait-relevant genes and mechanisms. The genes I omit, but this is true too. We can do this with messenger RNAs as well. Small RNAs are a component of heterosis formation in maize with an indicated overall negative effect, and the pericentromeric 22 nucleotide small RNAs likely suppress heterosis in maize. And I think for breeding, for hybrid breeding, these findings might have a potential to aid in defining heterotic groups, possibly even in other species or other crops. With this, I like to thank all people who contributed, and yeah, I want to thank for funding you very much for your attention, and I'm happy to take questions. The same trends, but the population was too small to significantly identify or associate a large number of small RNAs. There are few we find, but it's not enough. The best paraheterosis is a smaller value, and for this we couldn't address. But we are on the way to analyze larger populations.
Is that?
No, the graph was reduced to chromosome 5.
Okay.
And it's quite representative for all chromosomes. We define this by RNA-seq. We didn't look at this, to be honest.
Okay.
But yeah, for sure. I mean, from the hybrid analysis, we can tell that the sequences are there. I mean, we combine the genomes where we found the small RNAs expressed, and they should be there, but they are not expressed. A large number is not expressed in the hybrid, and there is new expression of new small RNAs are expressed in the hybrids. We analyzed this quite intensively with much higher expression threshold, and we see always a very similar picture. have some indications that the tissue we sample has indeed a quite strong effect. We analyzed ear tissue, so immature ears. Yeah, there is a lot of environmental effects on this, so I assumed always that our seedlings are fine for the prediction, but we see an increase in prediction accuracy when we use prediction accuracy for grain yield, when we use RNA data of immature ears. We grow these plants, it's the same population. I showed 21 inbred lines under greenhouse conditions in replicates, but yes, there is quite strong increase in prediction accuracy. Nevertheless, these data I showed for maize, they are with only 2,000 transcripts for the microarray, this custom microarray of seedlings. And even there we have an increase in prediction accuracy. So I think for practical reasons it's not wise to use later tissues, or yeah, the sampling should be as easy as possible, and we work on yeah, these controlled conditions and to sample really large populations under the same conditions, but our focus is there on seedlings still. I mean, we tested this effect only under in vitro conditions. I assume, or my hypothesis is that the programming in hybrids, the epigenetic programming goes on very early on, and there are indications from the literature that this is indeed the case, and then this programming is somehow fixed and maintained throughout development of the plant. That's why we induce in a very early stage, one day after pollination, and to get the inducer, this dexamethasone, into the tissue, into the cells, we need this in vitro system. I'm keen to create mutants and look for this under field experiments, but we need mutants for this to do it.